look back to the origin of this subject, it is the second oldest subject that is flourished in the late 16th century after the invention or maybe the improvement in the microscopic technique. Now, if you see from where the anatomy started, it is the first reference that you can get on based on anatomy. That is the link. Is the name of the scientist who has carried out work in 1607. So definitely the work must have been started around 1580 or 90. And this is the year of the publication because during that time there was no photography technique was available. Most of the things were hand drawings, and that is what the oldest book that is written in German. And this person has mentioned about the medullary bundles in Amaranthus. Okay, so that is the first reference you can say in the field of plant anatomy. Thereafter, maybe the microscopes were modified by Robert Hooke. I'll not go in much detail with that history. And he published his book Micrographia. And first time he mentioned about the cork, and that's the beginning of plant anatomy, actual what we say known to the science group. Then thereafter, of course, several people they started working with this, maybe around after 100 years, and major credit goes to certain people like D. Jusu, who has written a book again, Morphology, the family does Malpighiaceae. And means this is a person for the first time who used exclusively anatomical features for study of Malpighiaceae family. Then another person is West, who has studied the what we say anatomy and compared with all different groups of plants which are having vascular system like pteridophyte, gymnosperms, and angiosperms. Then another person who has major contribution is D. Barry. That is in 1884 he published a book. Basically, this is again in a German language, but it is a translated form which is available online also. If you search, you may get the book also. Comparative Anatomy of the Vegetative Organs of Phanerogams and Ferns. Then Bureau. That is the person who has exclusively used anatomy in the plant classification and delimitation facts of various levels within the family Bignoniaceae. It means whatever the references I have mentioned above, that is whether Link, Robert, D. Jusu, West, or D. Barry, these people were basically plant taxonomists, and they were using this technique as an additional support to discriminate the family or to discriminate the taxa. But actual credit goes to Bureau, who has done exclusively anatomical work to delimit the genera and species within this. And then thereafter, there are several names which came into the existence, like Schenk is there, Pfeffer is there, Metcalf and Chalk is there, Chateau is there. There are several people. Will not go in much detail, otherwise it will consume a lot of time. Now this is what then another contribution by Solarder. That is again one of the major contribution in the field of systematic anatomy of dicotyledons. Now if you see overall, so initially this anatomical data has been used as extensively for a taxonomic tool, only for the means for taxonomic point of view. And then it, what we say major credit that is within this, then you have August Matthews, then Solarder's work is there. That is important contribution, maybe oldest. And if you look at to these books, most of the things you will find hand drawings only. And here also that is what the title page of the book itself I have displayed. The diagrams were very accurate, but that is what maybe the older anatomy. Now if you look at to the what we say specific in the taxonomic point of view, as I go back to the Link work, initially what he has done is he has used anatomy as a complementary subject because it is 1605. So that time no microscope. So it was just visually whatever you are able to see. And this is how what we have depicted here I am showing. These are our images of Convolvulaceae family. But this is what same technique he has used because hand drawings indicate similar diagrams in his book. And he used it for showing whether growth rings are there or Initially, the even successive cambia were treated as a growth ring because of lack of information, whether it is phloem or xylem. They were treating it as a growth rings only. Then another example of this taxonomic work, pure taxonomic work, is the Hallier, who has used in 1884 to describe the members of convolvulaceae supplemented with micromorphology of the stem. So this kind of micromorphology they were using initially. But if you see the core anatomical, what we say, application that later on started just as a pure science to understand the pattern of growth and structural variation within a group, because that time they did not had any commercial intention behind that. It was just a place to, what we say, explore the knowledge, and that was the anatomical application during that time. Then to identify the genera or neighboring families in absence of reproductive structure. These are what we say, even today we use this application. These studies were just having the quest for knowledge, no any commercial interest. And since the commercial interest is developed, this subject has gone down, and maybe almost in Indian scenario, that is almost, you can say, wipe out. Very few people are there who are working on plant anatomy. However, as the information was generated, anatomy became one of the important science in identification of timber by forestry people. And even today, it is one of the important branch in timber market. Of course, we don't have this timber identification practice in India. Only few places are there like FRI Dehradun and Wood Science Institute, Bangalore, where you will find this identification. Otherwise, if you go abroad, maybe like Germany, France, or US, there people work as a consultant for sawmill people or timber traders for identification, as we do here for taxonomy. Okay. And they are the, what we say, the authorized people. They have the license and they can issue the authenticity certificate. That is what business is there, and that's the reason students they get the degree engineering in forestry or engineering in systematics. So that is what the branches they are having. Now, looking to this timber market success since ages, more than 200 years, this has been used in identification of wood. So we even started applying same technique to identify the fragmented material, that is drugs, medicinal plants, and which is still used in pharmaceutical science on the name of pharmacognosy, which is again the same scenario about the subject. This is what I'm mentioning here because at least somebody should get inspired and keep this subject live. That's the intention. Even I am today here to talk with you. That is what my intention, because the subject is diminishing in the country, and but real fact is, suppose FRI last scientist is working, that is Sangeeta Gupta. If she retires, then FRI even there is no expertise available in wood identification. Now, why it is important that we'll see later on. Then, after what we say taxonomy, with the development of various techniques and microscopy, it was extensively used in plant pathology to understand the entry mechanism of the pathogen, damage caused by the pathogen, and what is the plant response. Okay, so that was the major application during earlier days. And everybody knows Catherine Isau. Basically, she was a pathologist. Her duty, or what where she was working in Wisconsin, her duty was in agricultural university, that is agriculture and forestry, and she was expert in anatomy plus using anatomical techniques in detecting the pathogens and their role and how to control them. And gradually, then that subject developed a lot by Catherine Isau. She has the major contribution in general anatomy. Otherwise, if you omit Catherine Isau, then most of the work goes to the commercial point of view, that is in timber identification only. Now, recently, what is happening in the anatomy? It has several applications due to the development of recent techniques of sectioning, staining, sample processing, and even micro sampling methods. Now, here I'm mentioning about some techniques, that is recent techniques of sectioning. Means earlier days sectioning was different. They were using direct microtools, but now you have various parameters or various methods by which you can embed the sample depending on the hardness. Even the embedding material can be chosen. There are several techniques available online. Even you can search it. Then even microscopic methods. If you look at maybe light microscopy, bright field microscopy is there. Then you have fluorescence microscopy, dark field, and nowadays you are having confocal. Beside this, you are having electron microscopy, for which there is a sample processing. Not only that, even you can nowadays detect minor quantity of proteins also and DNA also by using this technique, particularly fish technique or fluorescence technique. And that's the reason it is used extensively in plant physiological studies and even developmental studies also. Now, last method I have mentioned, that is micro sampling. Now, micro sampling is nothing but when you say plant anatomy, usually it comes into the mind that you are supposed to cut the entire branch, cut the entire stem, or maybe bring the entire leaf, or maybe you are causing a harm to the tree or to the particular plant. But there are people, those who think even about the uh, pain felt by the plant, and that's the reason there is a micro sampling technique which has been developed, where you can extract only sample, maybe few microns, in a few microns, maybe less than 1 millimeter square millimeter. So that techniques are available, but of course quite costly, and at least in India we don't have that. And that is what undestructive techniques, again another technique where you don't have to take the section, you can directly observe any specimen. Only thing is it should fit into the machine. I will talk about the machines also, how it is observed, because here you need not to cut, you don't have to use the blade, you don't have to use any microtome. And those who are having very bad sectioning hand, for them it is a good news. Now it is also used maybe for correlating with the evolution, that how these organs or different tissues have been evolved and what is their role in the plant development. That is what uh, another application you have. Then there are several recent applications here. That is on left side. I have shown you maybe a phylogenetic tree that is not generated by using molecular data, but purely it is using anatomy. Okay, and that comes from the work Brazilian group from my colleagues only, Veronica, and her students or her research team. There are several families they have worked out, and using anatomical characters, they have generated this phylogenetic tree, which is almost, you can say, 99% matching with your APG classification keys. It means it is that accurate, but only thing is we should be well acquainted with the software which we are using. Otherwise, because it is a artificial intelligence, there are chances that it may commit the mistake. Unless we understand this, uh, using this technique also harmful for the subject. Now, where anatomy is used basically, that is illegal timber harvesting and trading. Everybody knows as a biology student, there is a lot of illegal cutting of the forest trees from the forest. And once it is cut, these people remove the bark. And once the bark is removed, then there are very few specialized people who can identify what the tree is. Many times people they cheat with the government also, because they purchase some barren land close to the forest. They undergo for agroforestry, cultivate the trees. They cut 10 trees from their farm and 50 trees from the forest, because they have the permission of cutting the trees growing in their farm. But this is how they do. So here you can identify whether the plant is grown in a farm condition by providing fertilizers, water, irrigation technique, or it is naturally growing. Second thing, even you can identify the species, whether the entire timber belongs to the same species or different species. Then wood identification, that is unknown origin. because many times it happens that the wood is cut, brought to the market, but what exactly it is? Many times, maybe even the sellers, end sellers, that is sawmill people, they sell something else on some other plant's name. Say, for example, if I remember maybe 20 years back, Melina wood was used for as an adulterant with the teak because of similar grain structure. But if you look at to the durability of the teak and Melina, there is a vast difference. But as the time changes and maybe the thinking of the human being changes, there are changes also. Now what has happened is at current scenario, Melina wood is more costlier than the teak because of its aesthetic value or it is related with the religion. Maharashtra, I don't have much experience about the people or their maybe uh, belief, but here in Gujarat, what they do is they consider that if you have a melana tree in your uh, maybe what you call in front of your house, or maybe you have a temple, ज्याला आपण देवारा म्हणतो, wooden temple prepared from this melana tree, that is good for worshipping the god, and that's the reason the demand has increased. And now here, as compared to teak, melana is costlier. Then plant pathology, that is host pathogen interaction, particularly in agricultural crops. Developmental biology, that is what we are doing nowadays. Molecular studies, we are altering the genes, we are overexpressing or maybe down regulating the genes, and what is their effect on wood structure, because many times wood is one of the important components in a human life, starting from the birth till death. You have the application of wood. Birth in the sense, once the baby is born, the first what we say your hanging basket, that is of wood, and the last where you go, the body which is burned, that is again used by using the wood. That's the reason I say birth to till death. Wood is our companion. And then taxonomic affinities and evolution of the plants in different groups. That is what the left side diagram depicts. Then ecophysiological adaptation of the plant. Now we are also facing the problem of what we say the increasing salinity and maybe the increase in barren land because of over what we say irrigation. So what are the alterations in the plant? That is ecological and physiological adaptation. That studies are conducted using this. Then stress related studies, various stress conditions, whether it is pollutant or maybe water type. Then indication of fruit drugs obtained from identification of fruit drugs, that is pharmacognosy. Forensic science, that is to solve the criminal cases. Now, criminal cases, we are totally unaware about this in India because our the people, those who are commit a crime in India, still they are illiterate, they are not educated, they are not that much scientifically developed. But if you look at to the well-developed countries, there even these criminals they are using modern techniques for killing or maybe what we say doing the murder. Say for example, using a wood as a knife, and once it is used maybe for poking purpose in the body, food can be thrown within 24 hours, your fingerprints will disappear. If you are using maybe a metal knife, there are chances that your fingerprints may live for few weeks or few months. But once the timber is thrown in moist place, within 24 hours, bacteria will attack on it, and whatever the fingerprints are there, that will be what we say disappear. That is how there are few cases have been reported in US, France, and Germany. Then there is another application in forensic science, that is charcoal, identification of charcoal. Because government all everywhere, even though the biodiversity board exists, there are certain trees all over India that can be cut without anybody's permission. You can burn it, make a charcoal, and you can sell it in the market. But what people they do is, on the name of permitted species, they cut even the permitted species which are not in the list. And once it is burned, means ultimately you have the charcoal. So even suppose a forest department or somebody authority or CITES people they catch you, they should have the evidence that suppose a X tray like eucalyptus or maybe say this Prosopis juliflora. is permitted for cutting, whether the charcoal belongs to eucalyptus or prosopis or something else, that can be used by using the anatomic identified, that can be identified using this charcoal method. And of course, archaeological studies also, because if you see the Indian history or mythology, we have very old temples also, but unfortunately, we never bothered what kind of wood is used, and that is what the studies on Indian ancient plants is carried out by one of the German lady. She is working in India with FRI group. She collects the plants from different wood samples from different temples, as well as maybe the mosque also, and what we call from the Girjagar or what we call church, or even Buddhism temples, what they are located in northern India. So that time, what kind of studies were carried out? This wood were used because maybe in sixth century or seventh century, what kind of wood was used for construction purpose? We don't have any information. That is what kind of studies people they do. That is all the applications. Only thing is, you should decide where you want to work or decide the topic, which topic you want to take, because whatever here on this slide I am representing, all of them, they are the application oriented topics. Now I'll just shift to the ancient technique, these ancient sculptures, because nowadays there are various groups which are working in various countries on the ancient sculptures, and these are the images which are of the wood sculptures in maybe various museums. Say, for example, like this National Museum in China, Germany, Belgium, and their images I am showing here. And this is nothing but it is the presentation of one of my friend who gave me the slides for this. He is a Japanese fellow, works in Nara Research Institute, Japan, and he works on this ancient world works. And you can see that these are the images or maybe the statues, you can say, which are of 5th and 6th century. And naturally, when it comes to the ancient sculpture, nobody will allow you to remove the wood sample. And that is the reason here the micro technique or maybe the micro sampling technique comes into the picture. Now, all these images here I'm showing on the upper side and lower side, three images are there which are showing the back from where the samples are obtained. But of course, you need a influence. No museum will permit you to obtain the samples, and naturally, you cannot make a big hole. But at least few millimeter wide samples you can take. This is how they sample the statues, so that even the religious people will not get sentimental hurt, because they cannot see from the front side that the image is damaged. Image on front side remains as such, but base portion back side you can obtain the sample in a very small quantity, and then arrow I am showing, you can see hardly you can recognize that the sample is taken out from the sculptures. And this is how maybe uh, the work which is carried out. Now here you can see that Bodhisattva. I think you may be aware about the Bodhisattva. Who was the person? Is there anybody who is having information? Hello? Anybody?
Anybody
Anyway,
can identify?
maybe in the sleeping mode. But I'll just say Bodhisattva. You can search out this name, put it on Google, or maybe if you are having a habit to watch a movie, maybe you can see that Chennai versus China or China versus Chennai, something South Indian movie. There you will realize who is the Bodhisattva, and he is treated as one of the major saint in China, and he went from South India to this China to prevent one kind of epidemic.
It's Bodhidharma, sir. Bodhidharma.
Bodhidharma, yeah, okay. But again, the another cases of Bodhisattva that even you can check it out. So That is what the studies here, and that is the reason the statues are there. And that samples what these people were using in 907 or from 907 to 1125 century to make these statues. And here you can see that the samples were obtained, they were sectioned, ordinary sectioning only, no staining, nothing else. And wood was identified using these techniques. Then this is again another case of Buddha statue, 6th and 7th century. These are the statues which are from various dynasties, either king or maybe the major rulers. Now I'll come to the non-destructive method. Here what you are supposed to do is you are not cutting the, what we say, the sample, or you are not getting the sample from the statue. But only thing is you should have the permission to bring the whole statue, or you should have the capacity to carry whole instrument in the museum. You can put the statue in front of the instrument, and like our X-ray photography, what we do for our bones, same way you can use this machine for taking the pictures, and this is how you can get the sequential pictures, and all these pictures can be compiled together and you can build an image, and that image can be used for the identification point of view without causing any damage, not even taking a 1 micron thick section from the specimen. And if you observe this under microscope, if you enlarge this one of the circle, half circle what I'm showing here, that is beaded nature is seen. Individual image, if you see, it looks like this under high magnification. And these are the techniques by which we identify the wood. For example, here you can see ring porous wood, that is Jelkova. Right side you can see the diffuse porous wood. Then again here on the left side, lower image, that is oak, where you can see the wide rays and wide band of axial parenchyma. And right side you can see the gymnosperm, where the distinct growth rings can be seen. This is how we go for the identification. Absence of vessel is another criteria to identify the gymnosperm. And this is how they identified the wood used during that time. Now this is again modern, what we say, advanced one, because here you are supposed to use a open one, or maybe you are using a box which is smaller, but suppose your specimen is larger, you can put it within the chamber, it's like a refrigerator, and there you can use. Or if again it is larger than, then you are supposed to go for this open type technique. And this is how the images which are obtained using this technique. Now this is something new, synchrotron, that is X-ray microtomography. Now on the left side you are seeing a very large two circles. So these two circles are nothing but, this is the entire single instrument. Single instrument in the sense, on our side or maybe on the lower side you can see a small building. It is occupying more than 1 square kilometer area. But the processing requires a very long tube-like structure which is spread over the circle. And from one side it comes out from the building, makes the complete circle, and then enters again back for interpretation of the result. And this is very, one or two instruments are established, and one of them that is located in France. That is the photograph synchrotron. Here you can see. Here also, without sectioning or without damaging, you can just, the instrument is fitted in the building. You can carry the entire statue over there, put it in the instrument, and then it process through various circuits, diodes, and digital circuit, what we say, processing technique, and almost complete the circle of 1 kilometer. And ultimately, the data which is again coming back to the building, that is analyzed using computers. And this is what overall principle I have shown here on the right side. So that is what it is, maybe underdeveloped technique you can say, because it occupies the large area, that much area one should have. And the sections or maybe the photographs obtained from that is like this. You can see the photographs without damaging anything. This is purely only the rays passing through the wood that can be constructed on a screen and can be observed in this way, and that is sufficient enough for the identification of the wood. You can observe maybe TS or LS or RLS in all the views, you can observe the wood for identification point of view. This is all maybe you can say the recent techniques which are used. Now another method is that is gross anatomy, and everybody must be aware about the Metcalf and Chalk, that is 1950s volume, which is available in two volumes. The book is very big and having around 1700 books in each, 1700 pages in each book. So you can say a Bible of plant anatomy. Any plant you live, almost 99% plant description you will find that, and 1% there are chances that the description is unavailable to you, and then you have a lottery to go for publication in a very good journal. Okay, so this is what maybe what parameters they are used. That is same outline how it looks, whether it is having smooth nature, ridges and furrows are there. Then distribution and abundance of the types of tissues. Then parenchyma thickening, kind of parenchyma thickening. Then variation in endodermis and pericycle. That is what uh, important criteria which is used in separating Piperaceae, Asteraceae and Lamiaceae. Then you can use that is peak portion, which can be used for identification of the families like Amaranthus, Chinopodiaceae, Solanaceae, Loganiaceae, because in all these cases you have the medullary bundles or internal phloem. That is the basic criteria for separating out. Then arrangement and type of vascular bundle, that is collateral or bicollateral. Then shape and size of the sclerenchyma ridges, that is what maybe cucurbitics in the last right side image using confocal scanning electron microscope, laser scanning electron microscope. So this is how maybe you can apply the technique, stain section, unstain macromorphology, and right side you can see that fluorescence technique can be used in identification, then variation in the structure of cell of the same endodermis. Then when it comes to the leaf, what parameters are to be observed? That is kind of stomata, cuticle thickening, then types of cell present in the epidermis, because uniform cells, here I am showing right side uh, cell, that image, that itself is an indicator of monocot leaf. On left side you can see stomatal crypt, it is in xerophytic plants. Sunken stomata is found in xerophytes, but not in a pure one. Here where the water scarcity is there, that is maybe a physiological desert condition or physiological this one you can see uh, the adaptation to the plant. And then again on the left side you can see a very thick cuticle where the plant wants to avoid the rate of transpiration. That is what maybe can be detected on just taking the leaf section. And other deposits also like crystals are there, druses are there, or spheraphites are there. What kind of deposition is there in the leaf, that can be used in the identification. I'll be just giving a taxonomic touch here. Then here I'll showing the different groups of plants where the how the leaf looks now, or maybe the branches they look. Here Achiranthus aspera, you can see the medullary bundles they are present. Then Moravia Nyctiagenaceae, medullary bundles undergo secondary thickening. Then number of medullary bundles as in case of Celosia and Amaranthus. These are the what we say the things which can be used in identification. Then when it comes to the anatomical aspect, then what aspects are to be seen, that is leaf anatomy, petiole anatomy, nodal anatomy, stem anatomy and wood anatomy. Uh, Maybe a lot of literature is available. Not going much in detail because we have already lost 15-20 minutes in joining starting the work, our presentation. Then leaf anatomical parameters, here itself you can identify the plant itself on the kind of the plant belonging to which group. Now here on the upper side, maybe left side image, you can see it's maybe a cam plant where you can see the succulents and kind of vascular bundle and the mesophyll cells. Then left side second image, that is ficus, you already studied. Looking to the transfusion tissue, you can see that mesophyll cells and spongy cell, in between there are horizontally running cells. That is spongy tissue which you will find in cycadales. Then on the lower side you have the dicot leaf. On right side it is coniferals, that is spinous again, because in coniferals group you have the needle shaped leaf, and that becomes a very unique. And then on lower side you have monoparts, which can be identified on the basis of type of cells. Epidermal cells, you can see the large cells in both the sections. Then you can see what we call transanatomy. That can be the criteria. Even means a small fragment, you can identify all level of plants. Okay, I have not included here any bryophyte or pteridophyte. Even that can be distinguished. Then petiole anatomy, what you are supposed to see, that is position of petiole on the stem, where it is located, absence of stipules, petiole outline, number of layers of parenchyma in the cortex, vascularization of petiole, then distribution of perivascular fibers, number of traces entering into the petiole. These are the important parameters that can be studied for delimiting the genera or species. And this is how just the cross section I'm showing, that is from Lopez et al. In Manihot. All the species can be distinctly separated out by using the petiole anatomy. And that is what here just want to show you how the vasculature that varies from species to species within the genus also. Then here we have studied some 12-15 species of Bowenia. You can look at to the arrangement of vascular bundles and then cortical bundles. They vary in all the species. In some cases they are absent. And you can identify all the species which are found in India just using leaf petiole. That is, even there are no flowers, no fruits, because Bowenia is a very tricky genus having around 350 species, and therefore it has been what we say splitted into various tribes like Bowenia, Panera, then Phylostigma, and Chinchilla. These are the various tribes from India. We have only 4 tribes here, and all of them have identified using anatomical technique by using the petiole anatomy. And you can see in all the cases the petiole shape, arrangement of vascular bundle, distribution of xylem and phloem, then cortical bundles. Everything is very distinct. Whether present, absent, if present, how it appears, whether it gives a what we call uh, something wing-like portion or wing is absent. All these criteria by using the leaf morphology, basic leaf morphology, you can reach up to the species level and no need to have the flowers or maybe any reproductive structure. Then sclerenchyma, what kind of sclerenchyma are distributed within the leaf? That can be another criteria that can be used for the identification at species level. Then of course this you already have studied different types of sclerids, where they are, how they are distributed. Then what type of deposits they found in the what we call leaf or maybe stem or cortex or any part of the plant body. Then stomata is another feature which you are well aware, and maybe even you might have learned the different types of stomata. That is anemocytic, anisocytic, parasitic, then diacytic, tetracyclic, actinocyclic, and then you are having few more additions. Cyclocytic is there, hexacytic is there, or graminaceous. From whatever I have shown, some of them you may not have studied, maybe at graduation level, but this usually we use it at maybe masters and PhD. Those students who want to delimit the taxa using anatomical features, then their cell wall pattern, that is again very, that is straight, sinus, wavy, beaded. All these natures can be used for identification and how overall it looks in a section view or maybe in a fields or in a using SEM technique that can be used as a supporting method. Then you have the trichomes, various types of trichomes that can be used as a unique character. Then kind of glandular trichomes, whether they are pubescent or maybe they are orky or maybe spiny or having the glands, that can be used as one of the additional supporting techniques. Then nodal anatomy very frequently used, but not at the genus species level restriction, but maybe to the groups and family level. It is well used by different people and has been used successfully. And even today, many of the studies which are carried out at college level, they use this technique because very easy. Thus, you are supposed to draw the what we say the gross anatomy using pen pencil, and because of that, this technique is very widely used even at several places uh, in all universities all over the India. And this is what we say the type of nodes where you have the traces, kind of traces which are entering into the petiole and stem nodes. Then I'll talk about the grass anatomy. If you look at to the overall structure, you can see the range of variation within the convolvulaceae family. That is Jaquimontia, Meremia, then uh, another Jaquimontia that is a variant in the flower, blue and yellow color. Then Meremia is there. Then Cressa, you have Evolvulus, then Meremia vitifolia, then Emarginata, and all these. Maybe all these members they vary. Here I am just cannot label because these are what we say from different families like Malpighiaceae, first left one, then right second one is the upper row, second one Leguminosae, then Sargania, then you have Malpighiaceae, then again Leguminosae, then Polygonaceae, Convolvulaceae, then in again this one Sapindaceae family, and lower side last two images on left side they are again Sapindaceae. Then Malpighiaceae and right corner that is Bignoniaceae you have studied round stain with furrows and phloem ridges, what we call phloem wedges. So here this is another genus which is having exclusively square stem and number of phloem wedges may vary. Here it is 4, can be 6, can be 8. Like that the number varies in different species. And this is what different members by which you can use this topographic study for separating out the taxa or maybe the within the genus, within the species level identification. Then besides circular stem, you have various stem shapes like it can be a pattern. First one is on the left side, that is Bauhinia. Climber Bauhinia are there in India also, that is Scandens, Bauhinia scandens you'll find. Right side Bauhinia, what we call Madagarensis. Then you are having Rincosia, then Macarium, and then another one is another species of Bauhinia, that is right corner left, right one, lowest image. So this is how maybe anatomical features can be used. Now there is another technique of food identification that is in absence of even morphological features. You have suppose 1 square centimeter cubic centimeter of wood piece by which even you can identify wood up to the genus and species level by using these three different criteria: chemical properties, odor and taste, then physical properties, how it looks, then anatomical feature, macromorphology and micromorphology. And you have the standard reference like taxonomy, you have the herbarium. Similarly, in anatomy, we have the xylarium, and here these are not the books, but these are what we say the reference material of wood samples, which are cut in a specific way, specific size, like book size or A4 size, and that can polish can be kept in this way in a cupboard according to families. And this is how the raw samples they are stored, which are received for identification. And this is the person that is Peter Gasson who works on leguminosae of the world, and he has identified or maybe generated the whole data on food anatomy of leguminosae family, but not the herbaceous members, which are commercially important. That's the role of Xylarium. Commercially important species, they are interested because they are working in the forest institutes. So non-commercial, nobody prefers nowadays, including us, even university and college people. Everybody thinks about the application, but basic science is the mother of what you call your recent branches, and that is the subject which is neglected nowadays. Anyway, when it comes to the physical properties, that is grain of the food sample, how it looks after polishing, then whether how it gives the, if you cut it in longitudinal view, how it appears, that is grain and straight grain and irregular grain. That can be pattern, one of the pattern. Then brightness, how it gives the shining. These are the original woods. You might have seen the Sanmaika. This kind of Sanmaika you will find in market also, but these are the original wood samples, and Sanmaika depicts these woods because they are very costly, cannot be afforded, and that's the reason We go for that 500 rupees, 600 rupees sheet of Sanmaika to show the maybe what we say our furniture. Anyway, so it comes to the anatomical character. Then what are the parameters? Like when you say angiosperm taxonomy, you need to have the flower, inflorescence. So we study the type of inflorescence, flower, polypetalous, gamopetalous, then kind of aestivation. Then we enter in detail with the number of stamens, gynosium, whether apocarpus, syncarpus, how many carpels are there, then placentation. There you have certain parameters. Similarly, in anatomy also you have the parameter that is the axial parenchyma, whether they are present, absent, if present, what quantity, they are abundant or maybe scanty. Then their arrangement, whether they are gregarious, spread. Then porosity, that is porosity is the word used to the vessels, whether they are present, absent, if present, whether forming the distinct rings or maybe what we call ring porous, diffuse porous. Then rays, whether rays are unicellular or multicellular. Then within rays also you have different types. Homocellular rays are there, heterocellular rays are there, depending on the cell size, or whether pedomorphic rays are there. Then kind of tylosis, because these are what we say the deposits in the vessels. And then you have the growth layers, that is growth ring, by which you can identify. Just 1 minute, I have to log in another meeting. In fact, there is another meeting at 12 o'clock where the presence is required. So I'm trying to log in. So yet meeting is to be started. So now these are the parameters which you can use for identification of the species or maybe wood identification. Now when you say the growth rings, then there are different types of growth ring boundaries. There are different types of growth ring boundaries. Maybe they may be distinct, indistinct, or whether it is formed by the parenchyma cells or fibers, what kind of tissue is involved. And on the basis of that, various kinds of growth rings, because this is entire maybe 3 to 4 hours session to talk all these features. I will be just briefly running with this. Then here you can see that the growth rings, they are not distinct, indistinct. Then what kind of vessel pattern is there? That is what you can see here. Then ring porous wood, what kind of vessel distribution is there? That you can see, maybe various types in different species. Of course, this entire data is based on your commercial wood samples only, not for the shrubs and herbaceous species, because the data is available only for this, not for the herbaceous and shrubby species. Because botany, we have never given the credit, and this is what now this information is totally shared by the forestry people because they use it in identification. Now, if you look at to these images, you can see the growth rings are very different in all three samples, whether it is fagus within the species. You can see on this basis itself, we can separate out the species. Then here you can look at to the pattern in the left side. You can see there are few large vessels. Then there are few small vessels which are angular. Whatever the larger patches, somewhat uneven forms you are seeing, they are the larger vessels. They are not perfectly circular, but they are angular. They are having the angles, maybe hexagonal or pentagonal. While larger vessels, they are oval to elliptic and smooth edge. Okay. Then whatever the rest of the portion is seen, they are the fibers. So parenchyma, they are scanty, very few. Then next image, if you see, where you can see that the vessels, they are going in a strips. If you see oblique orientation in the photograph, You can see that they are going in the strip. That is what you can say a dendritic pattern, which can be used for identification. And this is how the pattern of vessel distribution may vary. Right side image, you can see the ring porous wood. Initially formed vessel, they are larger and then gradually the size is decreasing. Here it is diffuse porous, but you can see in a unique pattern the vessels are formed, maybe somewhat zigzag. Then when you look at to the parenchyma, then they are what you say, maybe aliform or maybe a confluent. Here you can see they are the vasicentric or aliform parenchyma where you can see two large circles are seen on the left side, three basically looking like eyes and mouth. That portion you can see the portion looking like eyes and mouth. They are the vessels and surrounding. They are the parenchyma cells which are arranged having a very typical arrangement. That is what aliform arrangement. On right side you can see they may extend and form the bands, tangential bands. That is what aliform confluent. That is the what we say the typical arrangement for certain members of Leguminosae. Then again, Physalpinaceae from the same family Leguminosae, you can see the vessel distribution in Cassia, or maybe on the right side, you can see the parenchyma, whether they are initial parenchyma or terminal parenchyma, and if they are covering, it's enclosing the vessel on the upper side or on the lower side of the vessel, or they are encircling the complete vessel. In both the images, it is seen very distinctly. This is how you can see the this vessels as well as parenchyma distribution. Now on the left image, if you see peltogyne, where you can see the parenchyma, they are abundant and forming a large band. But on the right side diagram, if you see parenchyma cells are relatively less, but they are also on the upper side, but their number is less. Okay, they are not forming a continuous band. So these are the patterns which are well defined and available on the Inside Wood website. Okay, Inside Wood, that is the website maintained by International Association of Wood Anatomy, IWA in short. And this is how the parenchyma, whether if they are paratracheal, means away from the vessels or close to the vessel, what kind of arrangement they are having, whether they are, and for each type of parenchyma, there is a technical term, what we are having in taxonomy. So these terms can be used for describing the wood structure. Then this is how the axial parenchyma, that is banded parenchyma, they form the continuous bands, distinct bands by which species can be identified. And this is all maybe in brief. I have tried to cover only the identification aspect here so that in brief I can give you the overall idea about where the anatomy can be used. Particularly here, if you talk, maybe a individual topic itself is a very interesting. If you concentrate just on petiole anatomy or if you concentrate only on vessel anatomy, there you can uh, maybe you will really enjoy. But the only thing is your sectioning should be perfect and staining should be perfect. Otherwise, most of the time, what happens is the sections which we observe, the sections taken by the student, they are not less than 50 to 60 micron in thickness. And the good size of section to be observed is 12, 14 to maybe 20 microns. 20 is also very thick, but 14 to 16 or 17 micron is the perfect size section thickness for identifying. And not only that, even if you stain such section, they take a very perfect staining because whatever sections here I have shown, photograph section which we have taken, they are of 16 to 18 micron thickness. That's the reason you get the uniform staining. The reason is what happens is when we take the hand section, they are ununiform. One side they may be thick, another side they are thin, and depending on the thickness, the lignin content uh, kind lignin will be also more, and lignin takes the safranin. So because of that, one side it looks dark red, another side it looks light gray or light red. And if you are using again the color combination, like we use safranin fast green or maybe safranin astra blue or maybe we use collodion blue, depending on the kind of studies. And here, whatever this last slide you are seeing, this is the natural stain. We are not stained the section. It is just section was taken, it was dried, allowed it to dry, and then photograph is clicked. What do you think? It is a dicot or gymnosperm? Vessels are there. So will it go to the dicot or gymnosperm?
Anybody? TYBSc students.
TY students, at least you should know the difference between dicot and gymnosperm. Here the vessels are very distinct. I'll show the first image itself so that you can see in a larger view. The same photograph I have used here. Are you able to identify?
Kunal, Tejas, Supriya.
They are in a doubt. And of course, the photograph which I have shown, they would have definitely given the wrong answer. Because here vessels are thin, so you can say it's a dicot. But basically, it is a section of netum.
Netum. Yes.
That is, Netopsida is having vessels.
Yes.
So this is all. If anybody has any query, maybe you can ask.
If you, the session is open for questioning. If you have any queries, you can feel free to ask. Or you can write it down to sir later on.
Just a minute, let me log in. Huh. Now you can. Shall I remove this so that?
Yes, 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 sir. You stop presenting. Yes. Thank you, sir. So, if anybody has any questions, you can ask. Feel free to ask to sir. Gurav, sir.
Yes, madam. Yes.
Sir, hello.
Hello. Yes.
Sir, thank you very much, sir, for your guidance, sir. We have seen a very good work on anatomy.
Thank you, sir.
And what you say about that sectioning and the staining methods. Sir, we want your guidance for that kind of a sectioning and the staining of such kind of a work. So is it possible for us to organize such kind of sections, such kind of a section for the skill developments amongst the students for the sectioning and the sectioning method, staining methods of the such kind of anatomical work, sir?
Yeah, that can be organized. Even we are planning to organize such, but the thing is, because of this pandemic, we are helpless here. That's the reason.
So,
Otherwise,
so-
for time being, no,
So
even
because-
all this technique, Vidya is well aware.
Yeah,
She studied under plant anatomy laboratory only in the our
because
lab
sir,
only.
we have seen a very nice section, sir. We have seen very nice sections through your lectures, and
Yeah, yeah.
whatever the anatomical peculiarities were there, that was so much clear that anybody can understood about those anatomical features by observing those sections. It was
Yeah. No, in fact, that was a limitation of the time. Otherwise, I was thinking I will start with the very bad sections taken by the students and then talk to the student. This is how you observe, and that's the reason you don't have interest in anatomy. Basically,
Yeah,
it's a wonderful science. Once you get the good sections, I think you will leave other subject. I was crazy
Yes.
for taxonomy. Basically, how I enter in this subject, I was crazy behind taxonomy.
Yes, sir.
But
Yes,
no
sir.
fellowship was available, so I thought, let me join, complete my PhD, then I'll shift to the taxonomy. But within
Because-
that 3 years tenure, I developed so much interest at the family level. I started working on Amaranthus initially, and that was the beginning. Then I kept taxonomy as a supplementary subject to my uh, discipline. Otherwise, it is other way. People study taxonomy, they use anatomy as a supplementary subject, but I use taxonomy as a supplementary subject to the anatomy.
Yes, sir. Because many times sir, we use morphological characters, but how we can use anatomical character for the species level identification? I think you have very nicely explained it, sir, with the help of some examples. And thank you very much, sir, for that, sir.
Thank you, sir. Thank you for giving me this opportunity. In fact, I would have been happy to meet the students, but unfortunately, because of this net,
Yes.
you cannot make all of them online.
Yes. Yeah.
Yeah.
Yes.
Otherwise, it would have been a nice time to interact with the students,
No
because
issues, sir.
we have the hopes that some young generation people, they may come to this subject, because see, ultimately overall, if you look at to the Indian scenario, maybe from the universities or non-commercial point of view, only 2-3 people are there in India who are working on this subject. Otherwise, once these people they retired, I think this subject will be permanently closed in this country. Same scenario for wood identification. Once Dr. Sangeeta Gupta retires from FRI, then probably there will be no wood identification in FRI, because the rates are very less there, 500 rupees per sample.
Okay.
Bangalore will be continuing, because the professional one, they charge 5,000 per sample. So they may continue, but as such, on literature, on paper, no much information is available about the Bangalore group. Otherwise, once these two institutes, if they stop identifying wood samples, because